This next category is the lipid category, and this includes fats, oils, and waxes. Most are insoluble in water. If you mix oil and water, they will separate out. Lipids are found in all of our cells. They are an essential component of our cell membrane. And lipids provide twice as much energy as carbohydrates, that is, twice as many calories per amount as carbohydrates. Lipids are, a type of lipid is a triglyceride, and these lipids are made up of fatty acids, so you can see this long chain of what are called hydrocarbons, lots of carbon and hydrogen, just like gasoline. And so a triglyceride has 3 long fatty acids bound to one glycerol molecule. This is the chief component in our diet, and this is how lipids are stored in our body. Here's a single fatty acid, the building block of lipids. It is called a fatty acid because it has an acid functional group on one end, the carboxyl group here. It is a long chain of carbons and hydrogens. This fatty acid is a long straight molecule. This means that it is saturated. This type of saturated fatty acid is most often from animal sources. So there's 2 types of fatty acids, the saturated and the unsaturated. The saturated has no double bonds. The unsaturated has 1 or more double bonds. 1 double bond would be monounsaturated. More than 1 double bond is polyunsaturated. So here's a saturated straight chain molecule. You can see this straight chain here. Whereas here's an unsaturated fat, and at the double bond, this single double bond in the middle of the molecule bends or is kinked. Plant fats are often kinked or unsaturated. They are better for our health. The glyceride is referring back to our first fat, the triglyceride, where there were 3 fatty acids attached to a glycerol molecule. You can have a monoglyceride with 1 fatty acid, diglyceride, or triglyceride. You don't need to know your different fatty acids, but we get these from our food. Triglycerides, we get these from our food. Eicosanoids, which include prostaglandins and leukotrienes, are chemical messengers in the body. Steroids, you've probably heard that term before, but steroids are compounds we make in the body from cholesterol. So our body first makes the cholesterol, or we consume it when we eat animal protein. Cholesterol is found in the cell membranes of all animals. It helps to stabilize the proteins. And then we have additional structural components of cell membranes. Lipids may act as chemical messengers. That would be steroid hormones, and they may be part of the cellular structure, such as in our membranes. Prostaglandins are kind of interesting. These eicosanoids, including leukotrienes and prostaglandins, are produced by cells. They're like localized hormones. They affect the cells nearby. Steroids are very large molecules. We'll see that they're made from cholesterol, but they have a very different structure than those fatty acids we've been looking at. Cholesterol has 4 chains, so that's all we call, or 4 rings, pardon me. This is what we call these structures here, the rings. This is 4 rings of cholesterol. So our steroid hormones, such as estrogen and testosterone, are made from cholesterol as a starting molecule. So you can see that the large molecules that contain 4 carbon rings would be cholesterol and hormones. I'll skip this classification of lipoproteins, but in our blood, the lipids cannot travel by themselves. They have to be bound to proteins. And you may know about, if you've had a fat panel done on your blood, you may know about LDL and HDL, low-density lipoprotein, high-density lipoprotein. The LDL is the bad chemical, the HDL is the good one. Here's another fat, is the phospholipid. Because it's called a phospholipid, it contains a phosphate group. The phosphate group is bound to 2 fatty acids, and it is a structural component of our cell membranes. The glycolipid is a little different in that it has a carbohydrate attached to the fatty acids, and this is found on the outside of our cells, in the membrane, but on the outside of the cell, and often is used for cell recognition. So both phospholipids and glycolipids that we were just looking at here are composed of fatty acids. With a fatty acid, one end of the fatty acid is hydrophobic, that is water-hating, and the other is hydrophilic, water-loving. These tails here are the hydrophobic regions, and the head up here is the hydrophilic region. So if you were to put these fats with the hydrophilic head and the hydrophobic tails into water, they would line up so that all of the tails were in the middle, where they are farthest away from the water surrounding that. So you can see all of the water-loving heads are oriented to the outside where they're going to be exposed to water. This is called a micelle. Which of these lipids functions as a chemical messenger for local cellular activities? That would be the eicosanoids. Which lipid is part of a cellular membrane? Phospholipids. Which lipid has a ring structure? Steroids. Which lipids should be eaten in smaller quantities as it is bad for health? Saturated fats. Saturated fats are usually from animal sources. Unsaturated fats from plant. The last category are the proteins. Maybe second to last category are proteins. Very abundant in our body. They have many different uses, structural uses. They may be hormones, they're antibodies. They carry fats in our blood. One difference between proteins and carbs and fats is that proteins contain nitrogen. And when we're processing proteins, we have to get rid of that nitrogen. That's why our urine contains high amounts of nitrogen. So proteins are long chains built up of amino acids. In the body, there's 20 different amino acids. You do not have to know your amino acids, but our typical protein contains about 1,000 different amino acids. So amino acids, we have to talk about why they're called amino acids. Well, part of the structure is an amino group, NH2. Part of the structure is an acid or carboxylic group, the C double bond OH. And the central, they sandwich the central atom here, but the rest of the amino acid is called an R group, and it varies from one amino acid to another. The simplest amino acid would just have a hydrogen there for that R group. So all have the same structural components, a central carbon, amino group, carboxyl group, and the R group, which is variable. When amino acids are combined or linked together through dehydration synthesis, they form peptides. 2 amino acids form a dipeptide. many amino acids would form a polypeptide, and the bond that holds those amino acids together is called a peptide bond. So anytime you hear the word peptide, it's in the protein category. It may or may not be a completed protein. So here we are going to combine two amino acids, glycine and alanine, in a peptide bond through dehydration synthesis. So remember, when you are synthesizing, you are doing so by taking out a water molecule. This bond is called a peptide bond. The structure of proteins is critical. It is their shape that determines their function. So we have to know the 4 different structures of proteins. When we first build a protein, it is just a straight chain of amino acids. The average protein has about 1,000 different amino acids. But as those amino acids are being added to the protein, to this chain, the chain undergoes structural changes. All of the different side groups on these amino acids start to interact, and the chain will either bend into what's called an alpha helix. It looks like a slinky, or it will fold into a beta pleated sheet. This is called the secondary structure. These are all held into position by a variety of bonds throughout the molecules. When these chains interact, let's say you have 1,000 amino acids, and the first 30 amino acids are the alpha helix, and then the next 30 amino acids are a beta pleated sheet with a different structure, and then the last 40 amino acids are another alpha helix. Those themselves will start to fold over and bend and interact, forming what is called the functional tertiary structure. That tertiary structure forms a glob. These are also called globular proteins, but it is the functional unit. Now, in the case of hemoglobin here, hemoglobin consists of 4 individual chains that fold and interact and come together into a very specific shape with those 4 chains. This is the quaternary structure, where you have more than one amino acid chain. This structure is not really delicate. It's held together by hydrogen bonds and other bonding interactions, but it can be denatured. That is, if you were to heat that protein up, it would lose its structure. The tertiary structure would uncoil and become the secondary structure. That would uncoil and unfold to become the primary structure. This is called denaturation, and extremes in temperature or extremes in pH can denature proteins. What is the monomer or building block of proteins? That's amino acids. And the name of the bond that builds those proteins is the peptide bond. What structure forms an alpha helix or a beta pleated sheet? Secondary structure.